and welcome to our webinar today, Addressing the Challenges of Membrane Protein Sample Preparation for Cryo-EM Analysis. My name is Edward Pryor from Thermo Fisher Scientific, and I'll be your host for today. Our first speaker, Dr. Mahfuz Rahman, is an R&D Staff Scientist at Thermo Fisher and will be presenting the overall workflow of single-particle cryo-EM sample preparation, specifically discussing the challenges of membrane protein sample prep and sharing strategies and best practices to improve the success rate of sample preparation and obtain high-quality cryo-EM results. Following his presentation, our featured speaker, Dr. Rosemary Cater, an Associate Research Scientist at Columbia University, will discuss an approach used to solve structures of small membrane proteins using cryo-EM, where she complexes target proteins with specific monoclonal antigen binding fragments to increase particle size and provide fiducials outside of the transmembrane region for alignment. She'll describe how her and her lab used this approach to determine the cryo-EM structure of the major facilitator superfamily domain containing 2A, which has a molecular weight of 58 kilodaltons to a resolution of 3.0 angstroms. We'll conclude these presentations with a Q&A session where the three of us will answer any questions you might have regarding the topics presented in today's webinar. Before we jump into Mahfuz's presentation, I wanted to quickly just set the stage for today's presentations. In early 2021, along with Rosie's lab at Columbia University, we published a meta-analysis of membrane protein structural biology, where we looked at structure determination techniques used across the last 10 years. In 2019, you can see by this chart, was the first year where the number of cryo-EM, or membrane protein structures determined by cryo-EM, eclipsed the number of protein structures determined by X-ray crystallography. What's really exciting to show is that in the years that follow, in 2020 and 2021, this number has significantly increased. And so as you could see, if we add the last 2 years of data to this graph, you could see that the majority of structures determined, the majority of unique membrane protein structures determined by cryo-EM significantly eclipses the number determined by X-ray crystallography. I don't have the data from 2022 yet processed to be put in this graph, but a quick analysis shows that this number continues to increase and that close to 90% of the unique membrane protein structures were determined by cryo-EM in 2022. So why this increase? Well, we're seeing that this increase is due to more cryo-EM accessibility, increased throughput of the experiment, and really the focus of today's webinar, improved sample preparation techniques for membrane proteins. So what's next? If we look at all of the structures in the PDB, Richard Henderson proposes that by 2024, cryo-EM will be the predominant method for all proteins deposited in the PDB. So where are we today? So at last look, in 2022, we're currently about 64% of the structures that were determined or deposited in 2022 were determined by X-ray crystallography, with about 34% by cryo-EM. And if we look at a recent tweet or a recent graph from Masakiwa, he shows that, yes, even at current trends, we are projected to hit this number in 2024, as predicted by Richard Henderson. So with that, just to set the stage, I'll turn it over to Mahfuz Rahman, who will be talking about tools and strategies for improving sample preparation for membrane proteins.
Hi, thanks Eddie for the introduction. During my PhD and postdoctoral training, I worked with several challenging membrane proteins. And based on my experience, I will discuss on the power of cryo-EM and some challenges related to the cryo-EM sample prep and a simple sample optimization method. In 2017, when I was in my 4th year of doctoral studies, there was a huge recognition in the structural biology field for developing cryo-electron microscopy. The Nobel Prize was shared among Dubuche, Frank, and Henderson. Dubuche developed a method to keep the biomolecules in solution for cryo-EM application. He flash froze the biological sample in liquid ethane, where the liquid water solidified in its liquid form surrounding the biomolecules and retained the same condition as in the solution. Frank developed an image processing method to merge cryo-electron microscopes fuzzy 2D images to produce a sharp 3-dimensional structure. In 1990, Henderson succeeded in using an electron microscope to generate a three-dimensional image of a protein. And following these discoveries, with the advancement in instruments and sample preparation methods, the desired atomic resolution was reached in 2013. Now researchers can routinely obtain high-resolution structure of biomolecules using cryo-EM. During my PhD, I was using X-ray crystallography to study the structural biology of an ABC transporter at RIKEN SPring-8 Center in Japan, the largest synchrotron facility over there. In 2018, I visited San Francisco to present my crystal structure at Biophysical Society. In that conference, I was hearing a lot of success stories of membrane proteins using cryo-EM. At that time, there was very limited scope of cryo-EM in Japan. In the same conference, I also met my postdoctoral mentor, Dr. Ryan Hibbs. So I moved to USA to continue working on membrane protein structural biology and to learn single particle cryo-EM. During my postdoc, I was working with the ligand-gated ion channel called nicotinic receptor. The picture says it all, and it took several years from my PhD to solve one crystal structure of this ABC transporter. And to solve three different conformations of this transporter, it took around 10 years from my PhD lab from three different members. On the other hand, in my 4 years of postdoctoral work, I determined 8 high-resolution structure of this ion channel in large conformational state. So this story is not only my story. The same story goes for most membrane protein structural biologists. So cryo-EM filled a huge gap in the structural biology of membrane protein and protein complexes. So why cryo-EM? The X-ray crystallography method is limited to detergent and lipidic cubic phase. And in both cases, the membrane portion of the protein are not available to make direct crystal packing. And also in the presence of detergent cause the high solvent content, which causes fragile crystals or low diffracted crystals. And also in many cases, we could see the anisotropy in the data. On the other hand, cryo-EM offers a structure in native-like state. Moreover, the crystals in the crystals protein are static or rigid. Although ligand soaking is an excellent method, but the crystal will break if there is any significant structural change. In the cryo-EM sample, the proteins are in solution and dynamic to adopt any conformations. This slide gives an overview of the single particle cryo-EM workflow. The sample creation includes the expression, extraction, and purification of the protein. And the protein biochemistry need to be optimized to ensure that the purified protein is functional. In the vitrification step, we will add our protein sample to the grids and flash freeze in liquid ethane using Vitrobot. The next step will be to screen the grids to look at the different parameters to decide whether to optimize the biological sample of proteins further or to go for data collection. And processing the EM images will produce a 3D map, which will be used to build the atomic model. In this process, every step is critical and important, and in many cases need optimization. So what does a native-like state mean? I will give the example of my postdoctoral work on ligand-gated ion channels. The membrane proteins are embedded in the lipid bilayer of cell membrane, and therefore the lipids are very important for their stability and function. First, I purified the protein using detergent. Then I removed the detergent and again reconstituted the protein back to lipid bilayer and checked their ion channel function using electrophysiology. After finding suitable lipids for function, I reconstituted the protein in those lipids in presence of scaffold protein for structure analysis. This is a 3D cryo-EM map in low resolution showing that the lipid nanodisk is surrounding the membrane portion of the protein, which mimics the native-like state. To study the membrane proteins, the first step, we must extract them from the membrane, which is typically done by using detergent, and Thermo Fisher Scientific offers various detergents, including DDM, LMNG, and OG, OTG, and digitonin. Among them, DDM and LMNG is also available in combination with CHS. We also offer some classical detergent like Triton, Breeze, and Twin. In my next slide, I will talk about the GPCR extraction and stabilization reagent. To support the protein purification, we have affinity resin and magnetic beads for various tags like the His tag and DYK, DDDK, which is commonly known as flag tag, and GST tag and for HA. For grid preparation, we have a training kit that includes almost everything to start with grid freezing, and that is also including a standard protein apophyretin. In my later talk, I will discuss on a buffer screening kit for sample optimization during vitrification. And we also offer the negative staining solutions. For vitrification, Thermo Scientific offer Vitrobot Mark IV. So GPCR are one of the important drug targets, and in many cases, the receptors are not functional after extraction and purification in detergent. After screening various detergent and stabilization agent, we offer an all-in-one solution to extract the GPCR, also ensure the protein is functional. The reagent demonstrates an excellent extraction profile from both cell and tissue sources. We also offer an antibody that could be used to stabilize the GPCR G protein complex in active state. Vitrification process is a part of sample prep challenge in cryo-EM. Even a purified protein with nice biochemistry can behave differently in a thin vitreous ice layer. Ice thickness is important for the resolution. A thin ice like this is desirable, but the thick ice will cause adverse effect on the image contrast and thus limit the resolution. Another major problem during vitrification is protein aggregation. Ideally, the particle should be monodispersed and separated from each other. So for a high resolution data, a nice particle distribution is ideally needed. Preferred orientation of the particles is a recurring problem in this field. The left figure is showing a 2D class of a data where the side view is only present. In an ideal data for high resolution structure information, particles should adopt random orientation. To solve this problem, multiple rounds of optimization is often necessary. Currently, sample optimization is mostly performed in a non-systemic way, like changing grid types or changing the blotting force or blotting time, or using different detergent for protein purification or different salt concentration. And we also add different detergent to solve the preferred orientation problem. And this optimization takes more days or weeks to optimize the proteins. To reduce this, we have developed a buffer screening kit with a broad range of pH and salt concentration, and they are ready to use. The kit contains 14 buffers, and 6 detergents selected from over 800 cryo-EM papers published from 2014 to 19. A white paper is available with a detailed method on how to use this screening kit systematically identify the best condition for vitrification for a specific protein. Please make sure to check this white paper. This figure is from the white paper showing that buffer screening was helpful to find the best condition for 20S proteasome. The impact on unsuitable buffer ranged from protein aggregation, pore particle distribution, or preferred orientation. Another example from the paper showing that the detergent screening was helpful to solve the preferred orientation problem for coronavirus spike protein. The screening workflow is very simple and only need one working day. It involves the use of VitroBot and microscope with the EPU multigrid software, which enables automatic data collection for 12 grids in a day. 2 rounds of screening is recommended. First round to select desired protein concentration and the choice of buffer. The second round is to select the detergent. In both cases, it is recommended to check the biochemistry with the added buffer or detergent. And also keep in mind to avoid glycerol, sugars, or high concentration of salt in the cryo-EM sample buffer, because those compounds can have adverse effects on the vitrification or image quality. Apoferritin is being widely used as standard protein for single particle cryo-EM. The application of apoferritin includes microscope calibration, workflow validation, method development. And we are happy to share that we are supplying apoferritin as ready to apply on the grids. The sample is 97% pure and consistently providing high resolution data. The highest resolution we obtained was 1.7 angstrom resolution. If you are new to cryo-EM and want to learn the workflow, it is recommended to start your training with apoferritin. To sum up, I have talked about the protein prep focusing on cryo-EM, and I discussed on the related challenges during the vitrification and a simple screening method for the optimization. Almost everything from my talk is available in our website and in the white paper. As a product, we have apoferritin standard, buffer screening kit, training kit, and negative stance. The presented works were done in a collaboration between Rockford, Eindhoven, and San Jose site of Thermo Fisher Scientific. I am based in Rockford site, and I would like to thank Joanna, Aaron, Barb, Kay, and Chris. All the microscopy work was done at Eindhoven site. From that side, Dimple, Atia, Natalia, Albert, and Eddie was involved in this project. And thanks to Roja and Weijing from San Jose site for mass spectroscopy support. I would like to close my presentation. Thank you everyone for your attention.
Thank you, Mahfuz, for that great presentation. Now we'll turn the presentation over to Dr. Rosemary Cater, who will be talking about her work on the MFS D2A protein.
Okay, so today I'm going to be talking to you about an approach that myself and a number of other people in the Mancha lab at Columbia University have been using to solve the cryo-EM structures of small membrane proteins using cryo-EM. So almost 75% of the human proteome is made up of proteins that are less than 50 kilodaltons in size. And despite them serving a vast array of important biological functions, they only make up about 1.3% of proteins within the EM data bank. And it's not because they're not interesting proteins, it's not because no one's studying them, it's just because they're still technically challenging to determine the structures of these proteins using cryo-EM. And this is for a number of different reasons. Firstly, purely because of the size of these, they have a low signal to noise ratio, and also because these proteins typically lack distinctive tertiary features that allow us to align particles in the data processing pipeline. It's important to note that these two problems are even worse for membrane proteins because a lot of that signal gets buried within either a detergent micelle or a lipid bilayer for structure determination, so it just sort of exacerbates the issue. So today I'm going to talk to you about how I overcame some of these issues using antibody binding technology to determine the structure of a small membrane protein that transports fish oil from the blood into the brain. And so we all have heard of fish oil, we all have been told it's good for us, but what exactly is it, and what does it do, and how does it get to where it's needed in the body? So fish oil is made up of 3 different types of omega-3 fatty acids, and the one that I'm going to talk to you about today is called docosahexaenoic acid, or DHA. So DHA has this chemical structure here. It's got a charged or polar head group and a long hydrophobic polyunsaturated tail. And it's found in really high concentrations in the brain, so it actually makes up about 5% of the total weight of a neuronal cell membrane. And so as you can imagine, it's completely essential for proper neuronal development, functioning, and aging. Now, despite this dependence that the brain has for DHA, it can't actually make DHA, and we can make a small amount of it in the liver, but we actually heavily rely on dietary sources of DHA in order to fuel our brain. Now, of course, to get this DHA into the brain, it needs to cross the blood-brain barrier. So just to remind you of what that is, it's a layer of tightly packed endothelial cells that separate the blood from the brain parenchyma. And it exists, of course, to protect our brain from neurotoxins. However, it also creates a major bottleneck in neurotherapeutic development. So in order to get DHA into the brain, we have these DHA transporters that are called MFSD2A that are highly expressed in the microvasculature of the brain, and were first characterized by David Silver and his team at Duke US in 2014. And what their team demonstrated is that if you knock out this transporter in a mouse model, that you get significant reductions of DHA uptake into the brain and severe microcephaly and phenotypes that are reminiscent of omega-3 fatty acid deficits. So as the name implies, MFSD2A is a member of the major facilitator superfamily, or MFS, of transporters. And we're really lucky that there are a lot of crystal structures of members of this transporter family that have taught us a lot about the mechanism that they use to shuttle solutes across the membrane. So we know that they are typically made up of 12 transmembrane helices, and that these are separated into 2 6-helix bundles, the N-terminal domain in blue and the C-terminal domain in red, and that they use what's called a rocker switch mechanism to shuttle substrates across the membrane. So you can see on the left that the transporter starts off in an outward-facing V shape, which provides substrate and co-transported ions access to a central binding site, and that once they've bound, there's a conformational change, and the transporter then shifts to being in an inward-facing conformation, where the substrate can then be released to the other side of the membrane. So while we now know a fair bit about how this family of transporters work, it's important to remember that the crystal structures that we had available are of ones that transport small and relatively hydrophilic molecules. The substrate of MFSD2A is completely different. It's got this large multivalent head group and this floppy hydrophobic tail. And so it's reasonable to consider that the mechanism by which this transporter transports this substrate may be somewhat unique, and the membrane itself might actually be involved in that. And so we were really interested in understanding the structural basis of this. So some of you may have just noticed that the substrate I just put up there for MFSD2A isn't just DHA, it's actually DHA that's been esterified with a lysophosphatidylcholine, or LPC, head group. And so this is a lysolipid substrate. And this esterification is completely essential for uptake into the brain. However, MFSD2A can transport a number of different lysolipids. You just need to have a multivalent head group and at least 14 carbons in the tail, and preferably some degree of unsaturation. So going into this project, we didn't have a lot of information about how this transporter works. We did know that in the blood, LPC-DHA is carried around by albumin, and we hypothesized that it gets inserted into the outer leaflet of the membrane, and then that MFSD2A flips that lipid to the other side of the membrane in a sodium-dependent fashion. So as I said, I'm really interested in understanding the structural basis of this transport, not only because it serves a really important physiological function and it's very biophysically interesting, we don't know a lot about lysolipid transport, but also because if we can understand how MFSD2A transports LPC-DHA into the brain, then we can use this information to develop neurotherapeutics so they can hijack the system and enter the brain in the same fashion. So starting off this project, I actually started it in 2017, which was a while ago now, and back then I chose to start doing it using crystallography because this protein's only 58 kilodaltons in size, and so not really in the range for cryo-EM. And because this is a membrane protein and they're often quite difficult to express, the way that I started off this project is by screening expression of a number of different MFSD2A orthologs from different species that are have a good sequence similarity to that of the human, with it fused to a GFP tag to see which ortholog is stable and high expressing. And as you can see here, some express well, some don't. And from this, we moved forward with the MFSD2A Gallus-Gallus construct, as we could see that it was our highest expressing construct. We then went forward and did a detergent screen, so where we screen stability of our protein in a range of different detergents. And this is because, as you can see here, different membrane proteins are more stable and can have their structure determined in a range of different detergent environments. And so in order to do this, we then took our MFSD2A Gallus-Gallus and tried it in a range of different detergents, and it actually tolerated a lot of them fairly well, but that with DDM-CHS was the best and the sharpest, so this DDM supplemented with cholesterol hemisuccinate. And so we moved forward with that. Of course, we wanted to check that this transporter was functional, which we first did. And once confirming that, we then went and amped up the expression of this protein using a baculovirus system. We optimized our purification strategy. And from that, we went and laid some crystal trays. However, we never got any crystals, which is not an uncommon story in this field. And our first hypothesis was that this is because this protein actually has two glycosylation sites, and that can inhibit crystal formation. And so we then went and mutated out these sites, but that actually, those mutations actually reduced our expression of this protein quite substantially, to a point where we were pretty pessimistic if we'd ever be able to get enough protein for laying crystal trays. And so it was at this stage of the project that we started to consider if maybe cryo-EM could help us, because we know that over the last 10 years, the usage of cryo-EM to solve membrane proteins has dramatically increased to a point where it now overtakes that of X-ray crystallography, and that the average resolution of these structures is improving every single year. In addition to that, we also know that the size of the proteins that we can solve using membrane proteins, this is for, by the way, using cryo-EM is decreasing every year. So in the light blue and the dark blue down the bottom, you can see that proteins beneath 200 and even 100 kilodaltons are being solved with increasing propensity every year. So with this, we decided to give cryo-EM a shot. And another reason why we were really excited to do that is because it gave us the opportunity to reconstitute our protein in a nanodisk. So that's a sort of a nanodisk of a membrane bilayer that's surrounded by an amphipathic membrane scaffold protein. And so it sort of is a membrane mimetic. And you can see here that it's a somewhat popular choice, still not the main choice, but a lot of people do this. to determine membrane protein structures using cryo-EM. And for us, it was really important because remember, our substrate is a lysolipid, and we predict that the lipid bilayer may be involved in the substrate transport process. And so this was great for us. And so after optimizing this reconstitution process, we then made some grids of the protein and did some screening. And while this is just a screen, it's not a high resolution collection. When we saw this and we didn't see any transmembrane helices, we see essentially nothing sticking out of the membrane. Essentially it looked like a UFO to us. And so we were pretty pessimistic that we'd be able to get any high resolution information from these particles. And so it was at this stage of the project that we started to collaborate with the lab of Tony Kozyakov and his postdoc Satchel at the University of Chicago. And what Tony's lab has developed is this library of synthetic fabs, and that using phage display, you can screen your protein against this library to pull out ones that bind specifically to your protein of interest. And so this does two things to help with structure determination using cryo-EM. First of all, it bulks up the size of your protein. So we've gone from 58 to 108 kilodaltons in this case, which gives us more signal, but also it gives us an extra membrane feature to align our particles here and tell us, which way the particle is actually facing, what way is up, what way is down, and then align our particles more efficiently. I just put this slide in here just to point out that using antibodies isn't the only strategy for giving yourself a fiducial. People also use nanobodies, or as you can see here, there are a range of different strategies to do this, which has been reviewed really nicely by Wentic et al. In current research in structural biology just last year. And so here you can see there's a number of different strategies, including the NAbFab system, the megabody system, the Lego body system, and a range of target and fusion strategies to give yourself an opportunity to get a fiducial to get you that high resolution cryo-EM structure you're after. So after we were able to confirm complex formation with our FAB of choice, we then had performed some screening and then had a high resolution a collection on the Krios microscope. And from that, we were able to get these 2D class averages, which we were really excited about because as you can see here, you've got individual transmembrane helices within the nanodisk cloud and the FAB sticking out into the soluble space in lots of different orientations available to us. And so from this, we were able to obtain a 3 angstrom reconstruction of MFS D2A, and you've got the N-terminal domain in blue, the C-terminal domain in pink, and the variable domain of the fab up here on the outside of the membrane. And into that, we were able to build our structure of MFS D2A. As you can see here, it has a classic MFS D2A, it has a classic MFS fold, so 12 transmembrane helices separated into 2 6-helix bundles. And here you can see it's in this upside down V shape. So that's the inward facing conformation. So you've actually got this large intracellular cavity inside, which harbors a number of interesting features that I'll just show you very quickly. So I want you to imagine that you're standing in the middle of that intracellular cavity and you're looking left and you're seeing everything contributed from the N-terminal domain, and then you're looking right and seeing everything contributed from the C-terminal domain. And the first thing you notice is that it's very amphipathic. So residues from the N-terminal domain are relatively hydrophilic or charged, and those from the C-terminal domain are more hydrophobic. So looking at that N-terminal domain in more detail now, we saw a couple of interesting features. So up the top of the cavity contributed from the N-terminal domain, we can see that there's this charged central region. So this bunch of charged or hydrophilic residues, and it's actually directly adjacent to the sodium binding site, which would provide an ability for the coupling mechanism. And we thought this site looked really perfect for binding lysolipid head groups of our substrate. Of course, we wanted to confirm that these were functionally important, and so we did that in collaboration with David Silver, and I'm not going to go through all of the data now, but we saw a number of these, especially this E312, was particularly important for transport. Looking at the C-terminal domain in more detail, we saw something really interesting. So we saw that there's this deep pocket that's lined by hydrophobic residues that I've highlighted in pink. So this is just two different views of that pocket. And when we saw this, we thought it looks really perfect for binding a lysolipid tail. And what was even more interesting is that we saw that within this pocket, there was a cryo-EM density that we couldn't attribute to that we couldn't attribute to protein. And so when we saw this, we thought, have we captured a substrate in a binding pocket that just came from our expression system, which in this case was insect cells? And so in order to determine that, we sent our sample to Michael Marty, and Michael's grad student, James Keener, performed native mass spec on this sample and demonstrated that I actually had a 515 Dalton molecule bound to a subset of the particles present within the sample. Now, 515 Daltons is a molecular weight of LPC-18-3, which we also know is a substrate for MFSD2A. And so with that information, we then built in that LPC-18-3 into the density. And as you can see, it fits relatively nicely and matches up with the chemistry. Of course, we wanted to make sure that these residues were functionally important. And so we mutated them and had a range of different effects. Essentially, if you change the bulk or the charge of the residue, then you abrogate transport. And so when we have this substrate bound, if we look at the middle picture, we can see that the head group is sort of poking out in between this gap where the lysolipid head group is sort of interacting with the phosphate head groups of the inner leaflet of the bilayer. And so it really looks like it's poised to be released into that inner leaflet of the bilayer. And so in order to investigate that, we started to collaborate with George Kalashvili. And what George did is he ran molecular dynamics simulations of this protein in a range of different conditions and demonstrated that in a sodium-dependent manner, the lysolipid could move through this gateway between TMs 5 and 8 into the lipid bilayer. So it's actually this lateral gateway that provides a substrate with access to the membrane and exit from the protein. And so from this data and more, we were able to propose this mechanism for MFSD2A mediated transport, which I'm not going to go through in detail now, but if you want to read more about it, then we published it in Nature in June 2021. And what's also really nice is that since we put out this structure, there are now a number of other MFSD2A structures that have come out subsequently, all cryo-EM structures, all ones that either use antibody binding technology, nanobody binding technology, or other extramembrane binding partners binding to this transporter that have allowed for this cryo-EM structure determination. And it's taught us a lot about how this transporter mediates uptake of lysolipids across this lipid bilayer. And this is all thanks to these sorts of technologies that give us a fiducial to allow for cryo-EM structure determination. And I'd also just want to point out that this isn't just for one transporter. In our lab alone, we've now used this technology to solve the structures of a range of other, even smaller integral membrane proteins, all to a relatively good resolution, which is fantastic. It's a really powerful technology. And with that, I'd like to thank my mentor, Felipe Mancha, for everything that he's taught me and everything in the lab and the whole team, and also for Ollie Clark for teaching me everything about cryo-EM data processing, all of our collaborators and our cryo-EM facility staff, and also to all our funding agencies. And thank you for your attention.
All right. Thank you, Rosie and Mahfuz, for those great presentations. Now we've come to the live Q&A portion of this webinar. So if you have any questions, and a few have already started coming in for our presenters, if you do have any questions, there is a Q&A button on the lower left-hand corner of your window. And maybe we'll just kick it off. First question maybe for Rosie. So let's see, let me just actually find these questions. There we go. So you, in terms of the fabs that you added to your protein, are there any concerns or issues with those disassociating from the protein of interest?
Hi, Eddie. Yes, there can be in some cases. These are high affinity. The majority of the time they're on the nanomolar scale. However, a range of, and we typically add them at a 3-to-1 ratio to our protein. So we are already adding them in excess. But you can get dissociation of the complex, as you can with any complex. We've found that adding sort of detergent molecules or antipathic molecules, because this is due to air-water interface issue, right? So if we add on some amphipol or some chaps or some glycerizic acid or fluorooctyl multicide, and those are some of the antipathic molecules that we found that can prevent dissociation of particles because of that air-water interface.
Great. Maybe one additional follow-up question to that, or a related question that has come in. So the structure you determined was done in nanodiscs. Does this always have to be done in nanodiscs in order for this strategy to work of adding Fabs to the target protein, or can it be done in other membrane mimetics?
Right. So for this screening platform, because they screen in Chicago, they screen the library against using phage display. And so in this case, we reconstituted into biotinylated nanodiscs so that we can immobilize them on the plate. So you need some sort of biotinylation there to immobilize it. They have also done it in biotinylated amphipol you can also get, and you can use that as well. But yeah, you need for this specific sort of this specific FAB approach, you would need it to be in nanodisc for the screening. But as to whether those antibodies that you obtain in nanodisc also bind in other mimetics, I think it's probably quite likely that it has. It's not something that I've actually tested myself, but I'd be really interested to do that actually, because sometimes it can be beneficial to not have your protein in nanodisc to get around some other issues.
Sounds like a follow-up question, but Ali will maybe pause and pass over to Mahfuz. So you talked a little bit about the apoferritin standard. And I know this is focused on membrane proteins, but there was a question that came in about the apoferritin standard. Do users have to do anything to that protein? Is there any pre-work that needs to be done before applying that sample to a grid?
Yeah, thanks, Eddie, for the question. So no, the short answer. And we optimize everything, including concentration and buffer conditions. So there is no issue of aggregation. So you can directly apply to the grades. Thanks.
Great. Let's see. Here's I think there's about 5 questions in this one. Let me see if we can parse them out here. Maybe I'll ask the first one to Rosie. When reconstituting proteins into nanodiscs, how do you choose the correct lipid composition and maybe even a step further, the correct lipid ratio for nanodisc reconstitution?
So that's a great question because it is an issue. And I know we always talk about how we don't need much protein for cryo-EM, but when you're trying to decide these ratios and the lipid composition, you really want to perform a screen. So the way that I do this is and obviously that's going to work better if you have, it's going to be more achievable if you have more protein to do the screen. Although we do it on smaller scale. Just using an HPLC, we try a range of different lipid molecules, a range of different MSP proteins at a range of different ratios. So we screen all of those different conditions because I don't think that there is really a simple answer. Every protein is going to want a different makeup.
Great. All right, still parsing apart this one question. The second part, I think, is to you, Mahfuz. Is the GPCR extraction and stabilization kit that you talked about applicable for other types of membrane proteins or other classes of membrane proteins?
So at first, we tested on mainly for the GPCRs, and later we tested for other membrane proteins. And we have data that shows that also did a great extraction profile for other membrane proteins. We are still working on it to optimize for many membrane proteins and to recommend it for all of the membrane proteins. But from my experiments, yes, it can be applicable for all the membrane proteins, but it depends on protein to protein.
Great. I like this question because I have a feeling that all three of us will answer this question differently. So I'll ask it, and thankfully I don't actually have to answer it. So the premise of this question is I would like to know how essential detergent exchange is during purification. And I'll ask this maybe to you, Rosie, first, and then to Mahfuz second. In your opinion, is it a good idea to use a single detergent for both solubilization and purification?
That's an interesting, it's going to be different depending on your protein, right? And actually we put out a review on this 2020, I think, one of those fun COVID projects, where we compared the different detergents, the different protein families. The one that I put up for you within my talk that had the different protein families and the different detergents that we use, that was for the structure determination condition, but we actually also analyzed the different detergents that we use for solubilization and how often an exchange is. So I think that's probably the best place to go and look for your specific protein of interest to get a guide as to where to go. Yeah.
So yeah, I'll let's see if my hypothesis holds true that we'll answer this all differently. Mahfuz, in your opinion, is it a good idea to use a single detergent for both solubilization and purification?
And that is a great question. And actually, it depends on the protein, but I will answer it differently that the lipids are very important for the membrane protein. And when there is a harsh detergent, that is the problematic. But also, if you want to extract nicely from the native tissue source or from the cell, you sometimes might want to use the harsh detergent to do the extraction better. And at that time, around the membrane protein, there is all of the lipids is surrounding. So there is little harm compared to the after you purified it. So after purification, yes, always recommend the mild detergent, but for the extraction job, if you want to use little bit harsh detergent, that is okay most of the cases, but it's depend on the protein to protein and how much is the intercellular domain, is flexibility of the protein. So it depends on many factors. But surrounding when in presence of lipid, little bit harsh detergent, I will not mind. For my postdoctoral work, I use Triton, that is very harsh detergent to extract from the native membrane. And then I exchange to DDM, which is milder detergent. So it depends on the protein, but it is no harm to do any detergent exchange from my opinion. Thanks.
Thank you both. And since I really can't help myself, I do have to throw an opinion on this. I think I agree with both of you that it absolutely depends on the protein of interest, but I also think another factor is that it also depends on the end sort of goal of what you want to do with that protein, whether it's structure determination by X-ray crystallography or structure determination by cryo-EM or functional assays. I think that has a huge choice in the final detergent that you choose to put your protein in as well. So yes, as I predicted, all three of us have slightly different answers to that question.
But I agree with all of us as well.
Yeah, right? All right, there are a ton of questions here. Thank you all for putting these in. We're going to try to get to as many as possible. So let's see. So maybe for Rosie, for the fab technology, are there other factors that should be considered in selecting the right fab besides what you talked about, binding affinity and changes of function of the protein? Are there other things that should be considered?
Yeah, so I mean, because often you do get multiple FABs from these springs. I've had up to 40 come through. And sometimes those FABs will bind to different epitopes. And so sometimes if you want to have it, for example, I've had one that bound in the cavity where I was predicting that my substrate was going to bind, right? So that's not really ideal if you want to get a substrate complex structure. And so in that case, you then go and do the epitope binning, and you can determine which one's binding to a different epitope and then move forward with that one. Yeah, I would also say that I haven't observed this myself, but I wouldn't be surprised if different FABs stabilize different conformations of the protein. So it may be, once you identify if they have different epitopes, then you may want to see if they stabilize different conformations. But yeah, in terms of rigidity and using them for structural biology, they're all on the same backbone and have been optimized for that. So it's more just from what I can think of right now, the epitope.
Very nice. Yeah, a ton of questions about the Fab strategy here, and I'm trying to combine them in my mind to, to at least condense them. So while I'm trying to think of that, maybe another question for Mahfuz. Let's see if I can find one. Oh, this one we answered. Sorry. So you talked about the detergent screening kit, or I'm sorry, the buffer, the Vitre's buffer screening kit. And there's a question here to summarize. There's a lot of components to it. Does someone need to screen all of those buffers and all of these detergents in that kit to get a result, or is there a limited protocol that can be employed?
And that's a great question. So yes, there is flexibility. So it's recommend 12 grids because you can screen 12 grids at once. But it depends on the protein and depends on the buffer of choice. So if you know that your protein is happy within these buffers, or have from the literature that this protein is not good in this buffer, you can certainly use certain buffer from the screening kit and certain also detergent from the screening kit. So you can choose based on your likings or from literature review. Thanks.
All right, I think another Fab question for you, Rosie. There's a ton of them here. Let me start it. So were you able to collect and process any data without a Fab bound to your protein in terms of getting good 2D classes?
That's a great question. So for me, I definitely, because dissociation is a problem, right? You get a large number of particles that also don't have Fab bound. And so we process those as well. They did not go to high resolution in this case. I also, and other sort of strange things can happen as well when you're using the nanodisk system, like multiple monomers can go into one nanodisk, and that can bump one Fab off or the other. So yeah, in my case, I haven't been able to. But I do know that there are other situations where you wouldn't need, if you've got a very sort of rock solid protein, or it's forming a dimer or tetramer or other sort of oligomer, then you might be able to get high resolution without the FAB. But for this protein, and I think for ones that have large amounts of flexibility or large conformational changes, it's a little more challenging.
Great. Here's, I think, another question that we can probably pose to the two of you, and maybe also get different answers. So summary, this person is trying to reconstitute their membrane protein into peptidiscs after being extracted of detergents, but we see this, this process of being able to remove excess detergent from a solution. Are there other methods, or what are the best methods to sort of try to remove excess detergents after reconstitution in other mimetics? They mentioned dialysis, SEC, biobeads. Any others? Maybe we'll start with you, Rosie, that you can think of.
I've always just used biobeads with an overnight incubation with biobeads, and it's worked pretty nicely. All I can think of other than that is to have a detergent exchange before the extraction if you're having an issue trying to extract, but then that's always adding in another step, right? So that's gonna always introduce problems and time.
And maybe to you, Mahfuz, anything that you can think of besides the three that I mentioned in terms of removing excess detergent from a solution?
So yes, there is for another method for nanodisk formation is you just dilute the sample. So you added lipid and you added the scaffold proteins, and you dilute your protein in detergent without detergents adding buffers. So it will dilute, and the detergent will be diluted, and the protein will be more reconstituted into the lipid nanodisk. So that is another type of method. But I think BioBits is preferable because it does a good job, because it absorbs detergent by over the time. It's not a harsh process for the protein, and it's mild process, so little bit exchange between detergent and then lipid and then detergent little bit more. So it's not a harsh process. So, but yeah, there are some other methods also to check that. Thanks.
Great. Rosie, are you tired of answering fab questions yet? Because I got about
No, I can answer some more. I'm glad that people are interested in it.
So the question is, if the fab finalized for the structure determination affected the function and or stabilized it in a particular conformation, and then sort of as a corollary, were several fabs screened for function first before deciding on the final fab?
So the question is, are the fabs affecting the function and...
are they locking it into a particular conformation and or affecting the function of the protein?
Yeah, so I didn't do that myself, but I think it's a great idea, because if you've got a good functional assay set up for your protein of interest, and you know how to read out different conformational states or substrate binding or inhibitor binding or whatever, then I think that that's a great place to start to see if you can, whittle out those ones that are going to bind to your substrate binding site before actually putting them on a grid. I think that's a great idea.
Great. There are a ton of questions here. Let's see. So I think a follow-up question to something you mentioned before, Rosie, about multiple membrane proteins in one nanodisk. And the question is, how do you know if your nanodisk has 2 or 3 membrane proteins, and how does it affect what you see in cryo-EM in terms of resolution of particles?
So when I did see, in this case, I could, first of all, the nanodisk size changes, right? Because they can expand or contract, which is what allows this to happen in the first place. And so I could see multiple, they weren't highly resolved with nice transmembranes like I showed you before, but you could see multiple protomers within there. And in this case, I don't believe that this is a real dimer or tetramer or whatever the case was, because of it not happening in not getting that same size eluted when I have the protein at, say, a lower concentration, or in a detergent screen beforehand, it came off at a retention time that's more reminiscent of a monomer. But yeah, you can pretty much just see it by eye by the size of the nanodisk, and you can see I think in that paper that we put out, that I actually did include the different 2D classes, the ones that have a fab bound, no fab bound. So you can take a look there in the extended data if you want to get an idea.
All right.
So we added with that unlike the MSPs, the sapogene actually can get any radius they want. So in case of my experience with my protein, that there was in native membrane, they are as a dimer. So they can actually, in total, it was around 500 kilodalton protein, big protein. The sapogene can take them as one. And the resolution, it depends on how direct binding or how many interface of interaction they are with each other, or in simple, how flexible is their interaction. If their interaction is rigid, the resolution will be higher, and it's always help to make a bigger complex for cryo-EM. But if they are flexible and rotate with each other, then it will be difficulty with the alignment, and it will be difficult to get high resolution data.
So we are nearing the top of the hour, and unfortunately, there are still a lot of questions left to be answered. So I will mention that anything that we did not have time for during this Q&A session, we will answer offline via email. I do want to take the time here to again thank both Rosie and Mahfuz for participating in this webinar, and really thank all of you. I think we had quite a number of people attend this, and probably the most questions I've seen come in from an audience. So it's just more reason that we would, I think we have to do this again in the near future. So again, thank you, Rosie. Thank you, Mahfuz. And thank all of you for attending this webinar. And I hope everyone has a great rest of their day.
Thank you, Eddie.
Yeah, thank you.